Hello, and welcome to Calm Science Nights. I'm so glad you're here with me tonight. Before we begin our journey into the hidden world of plants, if you find peace in drifting off to the soothing rhythm of scientific discovery, please consider subscribing to the channel. It helps us create more of these tranquil explorations together. Now, settle into your most comfortable position. Imagine yourself lying on soft moss in a quiet forest clearing, surrounded by ancient trees. Feel the earth beneath you, solid, patient, alive. Above you, leaves rustle in the gentlest breeze, and all around, though you cannot hear it with your ears, a conversation is taking place. Plants are speaking to each other in languages we're only beginning to understand. Chemical whispers through the air, electrical pulses through the soil, networks of connection beneath your feet more intricate than any human internet. Tonight, we're going to explore one of science's most beautiful revelations, that the green, silent world around us is neither silent nor solitary, but engaged in constant communication, cooperation, and even something that looks remarkably like intelligence. Over the next 100 facts, we'll trace humanity's journey from seeing plants as passive, unfeeling objects to recognizing them as active participants in complex ecological conversations. There's no need to stay awake for all of it. In fact, the plants themselves would approve of your rest. They understand the importance of cycles, of darkness, of the quiet restoration that comes with letting go. Just allow these words to wash over you like sunlight filtering through a canopy, dappled and gentle, carrying you peacefully into sleep. Close your eyes now, and imagine a single plant, perhaps a tomato plant growing in a garden on a warm summer day. To most observers throughout human history, this plant appeared to simply exist, passively photosynthesizing, growing slowly, without awareness or agency. But in 1983, two scientists named Jack Schultz and Ian Baldwin made a discovery that would begin to change everything we thought we knew about the inner lives of plants. They were studying how plants defend themselves against insects, working with willow trees and poplar trees in a carefully controlled experiment. What they discovered seemed almost impossible at first. When they damaged the leaves of one tree, tearing them to simulate insect feeding, not only did that tree begin producing defensive chemicals to make its leaves less palatable to insects, but nearby trees that hadn't been touched at all, also began producing these same defensive compounds. It was as if the damaged tree had sent out a warning, and the neighboring trees had heard it and responded. The mechanism, they discovered, was airborne. When a plant's leaves are damaged, it releases volatile organic compounds, chemicals that evaporate easily and drift through the air. These aren't random emissions but specific molecular messages, and other plants can detect them through receptor proteins in their leaves. When a neighboring plant receives these chemical signals, it interprets them as a warning of danger and activates its own defensive systems preemptively, even before any insect has touched its leaves. This discovery, published in 1983, was revolutionary because it suggested plants were not isolated individuals but participants in a chemical communication network. The scientific community was skeptical at first. The idea that plants could talk to each other seemed too close to anthropomorphism, too much like attributing human qualities to organisms that clearly lacked brains or nervous systems. But as more researchers began investigating, the evidence became overwhelming. Plant communication wasn't metaphor or wishful thinking, it was measurable, repeatable biochemistry. What made this even more remarkable was the specificity of the communication. Later research revealed that plants don't just release generic alarm signals, they can produce different chemical messages depending on what's attacking them. A plant being eaten by caterpillars releases different volatile compounds than a plant being attacked by aphids, and neighboring plants can distinguish between these signals and tailor their defensive responses accordingly. It's a sophisticated chemical vocabulary, with different words for different threats. But the story of plant communication actually begins much earlier than 1983. Those scientists at the time didn't recognize what they were observing. In the 1960s, a researcher named Gottfried S. Frankl was studying insect feeding behavior and noticed something curious. Insects seemed to prefer certain individual plants over others, even within the same species growing in the same conditions. Some plants were heavily damaged while their immediate neighbors remained untouched. Frankl suspected this had to do with chemical differences between plants, that some individuals produced more or less of certain compounds that insects found attractive or repellent. What he didn't realize was that he was observing the effects of plant communication. The plants that remained undamaged may have been receiving warning signals from their damaged neighbors and responding by increasing their chemical defenses, making themselves less attractive to the insects. The plants were already talking to each other, science just hadn't learned their language yet. The breakthrough in the 1980s opened the floodgates for plant communication research, but it also raised profound questions. If plants could send and receive chemical signals through the air, what other forms of communication might they be using? The answer, it turned out, was beneath our feet. While scientists were studying airborne signals, they were missing an even more complex communication network running through the soil, a network that would eventually be called the Wood Wide Web. This underground network centers on fungi, specifically, mycorrhizal fungi that form symbiotic relationships with plant roots. The word mycorrhiza literally means fungus root, and these partnerships are ancient, dating back over 400 million years to when plants first colonized land. The arrangement is elegant. The fungus grows into and around plant roots, extending thread-like filaments called hyphae far out into the soil. These hyphae are incredibly fine, much thinner than plant roots, allowing them to penetrate tiny soil spaces and access water and nutrients that roots alone could never reach. The fungus delivers these nutrients to the plant, and in return, the plant provides the fungus with sugars produced through photosynthesis. For decades, scientists understood this relationship as a simple trade, nutrients for sugars, a mutually beneficial arrangement between two organisms. But in the 1990s, a researcher named Suzanne Samar, working in the forests of British Columbia, discovered something that transformed our understanding of these fungal networks. The fungi didn't just connect individual plants to the soil, they connected plants to each other. Samar was studying how different tree species interacted in mixed forests, particularly Douglas firs and paper birches that often grow side by side. Using radioactive tracers, carbon isotopes that could be tracked as they moved through the forest ecosystem, she made a stunning discovery. When she labeled the carbon in one tree by exposing it to radioactive CO2, that labeled carbon didn't stay in that tree. Within days, it appeared in neighboring trees of different species, sometimes trees several meters away. The labeled carbon was traveling through the mycorrhizal network. The fungal hyphae that connected one tree's roots also connected to other trees' roots, creating a continuous underground network through which nutrients, water, and even information could flow. Samar found that this wasn't just passive leakage. The exchange appeared to be regulated, with larger, older trees often transferring resources to smaller, younger trees, especially when the younger trees were shaded and struggling to photosynthesize enough for their own needs. These large, old trees, which Samar called mother trees or hub trees, served as central nodes in the forest network, connected to dozens or even hundreds of other trees through the fungal web. They appeared to play a nurturing role, supporting the younger generation, and when these hub trees were cut down, the survival rates of younger trees in the area decreased significantly. The forest, it turned out, was not a collection of individuals competing for resources, but a cooperative network, with the oldest and largest members supporting the community. And so the hidden world began to reveal itself. Schultz and Baldwin's 1983 discovery that damaged willow and poplar trees release airborne chemical warnings, and neighboring untouched trees respond by activating their own defenses. We learned that plants produce specific volatile organic compounds for different threats, different chemical words for caterpillars versus aphids, allowing neighbors to tailor their defensive responses. We traced the story back to Frankel's 1960s observations of insects preferring certain plants over neighbors, unknowingly witnessing plant communication before science had the framework to recognize it. We discovered the mycorrhizal partnership over 400 million years old, fungal hyphae extending into soil spaces too small for roots, trading nutrients for plant-produced sugars. We followed Samar's 1990s radioactive tracer experiments in British Columbia forests, revealing that labeled carbon from one tree appears in neighboring trees of different species within days, traveling through the fungal network. We learned of mother trees or hub trees, large, old trees connected to hundreds of others, transferring resources to shaded, struggling younger trees, their removal decreasing survival rates of the younger generation. Each discovery revealing not competition but cooperation, not isolation but connection, not silence but conversation in languages we were only beginning to hear. The forest floor alive with messages, chemical and fungal, the green world engaged in constant dialogue. Feel yourself resting on that forest floor now, part of this vast network, connected to something larger than yourself. The trees above you breathing out oxygen while you breathe it in, the fungi beneath you weaving their invisible threads, the whole system pulsing with life and communication. Nothing for you to do but rest here, supported by ancient systems that know how to sustain life, how to share resources, how to communicate across boundaries. Let yourself sink into the soft earth, into the welcoming darkness, into the quiet conversation of the forest. As we continue our peaceful journey through the hidden language of plants, we discover that the underground fungal network Suzanne Samar revealed was just the beginning of a much deeper story about how plants exchange information beneath the soil. The mycorrhizal networks don't just transfer nutrients and sugars, they appear to carry warning signals as well, creating an underground alarm system that complements the airborne chemical messages we discussed earlier. In the early 2000s, researchers began conducting experiments where they would stress a plant in one part of a network, perhaps by exposing it to drought conditions or introducing harmful insects, and then observe how other plants connected to the same fungal network responded. What they found was remarkable. Plants connected through fungal networks showed defensive responses even when they were too far apart for airborne chemical signals to reach between them, and even when barriers prevented any above-ground communication. The only connection was underground, through the fungal threads. When one plant was attacked by aphids, For instance, connected plants would begin producing anti-aphid compounds within hours, despite having no direct contact with the insects themselves. The message was traveling through the soil, through the fungal network, faster than the aphids could spread. This underground communication appears to work through both chemical and electrical signals. The fungal hyphae can transmit chemical compounds from plant to plant, carrying warning molecules through their network of threads. But researchers also discovered that the fungi generate electrical signals, small voltage changes that propagate through the network when a plant is stressed. These electrical pulses travel much faster than chemical diffusion, potentially allowing rapid warning transmission across considerable distances. The speed of these electrical signals through fungal networks was measured in laboratory studies using sensitive electrodes. Researchers found that when one plant was damaged, electrical activity in the fungal network increased within minutes, and plants connected to that network showed defensive gene activation within 1 to 2 hours, far faster than could be explained by chemical diffusion alone. The fungal network was acting like a biological telegraph system, carrying urgent messages through electrical pulses supplemented by chemical confirmation. But the mycorrhizal network isn't the only underground communication pathway plants use. In 2013, researchers made another surprising discovery. Plants can communicate directly through their roots, even without fungal intermediaries. When roots of different plants grow close enough to touch or intertwine, they can exchange chemical signals directly, root to root, in a process that looks remarkably like the synaptic communication between neurons in animal brains. The mechanism involves exudates, chemicals that plants release from their roots into the surrounding soil. Every plant constantly secretes a complex mixture of compounds from its roots: sugars, amino acids, organic acids, enzymes, and specialized signaling molecules. These root exudates create a unique chemical signature around each plant, and other plants' roots can detect and respond to these signatures. When a plant detects stress-related compounds in a neighbor's root exudates, it can activate its own stress response pathways, preparing for potential threats. What's particularly fascinating is that plants appear to be able to distinguish between root exudates from their own species versus other species, and even between roots from their own individual clone versus genetically distinct individuals. Studies with sea rocket plants, which grow in beach environments, showed that when grown next to genetic strangers, they developed more extensive, competitive root systems. But when grown next to clones of themselves, genetically identical individuals, they allocated fewer resources to root growth and more to above-ground shoots, essentially cooperating rather than competing with their kin. This ability to recognize kin suggests a form of plant intelligence that goes beyond simple chemical reflexes. The plants are processing information about their neighbor's genetic identity and adjusting their growth strategy accordingly: cooperation with relatives, competition with strangers. It's a strategy that makes evolutionary sense. Helping genetic relatives increases the likelihood that shared genes will survive into the next generation, while competing with unrelated individuals helps ensure access to limited resources. The root-based recognition system also allows plants to detect whether they're in soil that's been previously occupied by the same species. Plants can sense the specific signature of root exudates left behind by predecessors, even after those plants are gone. In some cases, this triggers allelopathy, the production of chemicals that inhibit the growth of other plants. Black walnut trees, for instance, are famous for producing a compound called juglone in their roots and leaves. Juglone accumulates in the soil around walnut trees and inhibits the growth of many other plant species, essentially creating an exclusion zone where the walnut tree faces less competition. But allelopathy isn't always about exclusion. Some plants use root-exuded chemicals to facilitate the growth of beneficial neighbors, a phenomenon called positive allelopathy or plant facilitation. In harsh environments like deserts, some pioneer plants modify the soil chemistry through their root exudates in ways that make it easier for other species to establish themselves. These nurse plants essentially create favorable microsites that support greater plant diversity, even though this means accepting more competition for resources. The complexity of these root-mediated interactions suggests that the soil around any given plant is a rich information environment, full of chemical messages from neighbors, from microbes, from fungi, from the decomposing remains of previous generations. Plants are constantly sampling this chemical soup, extracting information, and adjusting their growth, their defense, their resource allocation in response to what they detect. The soil isn't just a physical substrate holding plants in place, it's a communication medium, thick with information. And so the underground conversation deepens, mycorrhizal networks carrying not just nutrients but warning signals, plants connected through fungal threads showing defensive responses to threats attacking distant neighbors, even when too far apart for airborne chemicals to reach. We learn these networks transmit both chemical compounds and electrical signals, voltage changes propagating when plants are stressed, electrical pulses traveling faster than chemical diffusion. Laboratory studies measuring these electrical signals found defensive gene activation in connected plants within 1 to 2 hours of a neighbor being damaged, the fungal network acting as biological telegraph, urgent electrical messages supplemented by chemical confirmation. We discovered that in 2013, researchers found plants can communicate root to root without fungal intermediaries, exchanging chemicals directly when roots touch or intertwine. We learned of root exudates, complex mixtures of sugars, amino acids, organic acids, enzymes, signaling molecules secreted continuously, creating unique chemical signatures. Other plants detect stress compounds in neighbors' exudates and activate their own stress response pathways. Sea rocket plants distinguish genetic kin from strangers, developing competitive root systems with strangers but cooperating with clones, allocating more to shoots, less to roots when growing beside genetic relatives. We found plants can sense root exudate signatures from previous soil occupants, sometimes triggering allelopathy, black walnut trees producing juglone that inhibits other species, creating exclusion zones. Yet positive allelopathy also exists, nurse plants in harsh deserts modifying soil chemistry to facilitate other species' establishment, creating favorable microsites despite accepting more competition. Each root system constantly sampling the chemical information environment, the soil thick with messages from neighbors, microbes, fungi, decomposing generations. Not just substrate but communication medium, the underground world alive with chemical conversation. Feel yourself resting on this information-rich earth, the chemical messages flowing beneath you, roots extending in all directions, touching, sensing, responding. The whole system intelligent in its own way, processing information, making decisions, adapting to circumstances. Nothing for you to direct or manage. Just rest here, supported by these ancient networks of connection and communication. As our journey through the plant world continues, we move from the hidden language beneath the soil to another remarkable discovery about how plants sense and respond to their environment, one that challenges our very definition of perception and awareness. In 2014, a researcher named Monica Gagliano published a study that sent ripples through the scientific community. She was investigating whether plants could learn, specifically whether they could habituate to harmless stimuli, essentially learning to ignore something that initially seemed threatening but proved to be benign. The concept of learning had always been associated with nervous systems, with neurons and brains, so the idea that plants might learn seemed impossible to many scientists. Gagliano worked with Mimosa pudica, a plant commonly called the sensitive plant because its leaves fold closed rapidly when touched, a defensive response that presumably protects it from being eaten or damaged. She designed an ingenious experiment where she repeatedly dropped the potted mimosa plants from a height of about 15 centimeters onto a cushioned surface. The drop was startling to the plant, causing it to fold its leaves, but it caused no actual damage. Initially, every drop caused the leaves to fold. But after just a few repetitions, the plants stopped folding their leaves in response to the drops. They had learned that this particular stimulus, though initially alarming, was actually harmless. Even more remarkably, when Gagliano tested the same plants a month later without any drops in between, they still remembered. They didn't fold their leaves when dropped, demonstrating that the learned response persisted over time. The plants had formed a memory. This discovery of learning and memory in plants without nervous systems forced scientists to reconsider what these cognitive processes actually are at their most fundamental level. If learning is simply the ability to modify behavior based on experience, and memory is the retention of that modified response, then plants clearly possess both, even without neurons. The mechanisms are different. Plants use networks of chemical and electrical signals within and between cells instead of neural networks, but the functional outcomes are remarkably similar. Building on this understanding of plant memory, researchers began investigating whether plants could make decisions, whether they could assess different options and choose between them based on circumstances. One elegant study examined how climbing plants decide where to grow. Climbing plants like peas, beans, and morning glories need to find something to climb on to reach sunlight, and they do this by growing in a spiral pattern, essentially searching the space around them for support. Researchers presented climbing plants with multiple potential supports at different distances and orientations. What they found was that plants didn't simply grow toward the nearest support. Instead, they appeared to assess the available options based on multiple factors: distance, height, thickness, stability, and then direct their growth toward the support that offered the best overall opportunity. When presented with a choice between a nearby short stick and a more distant tall pole, many plants chose the tall pole, presumably because it offered better long-term access to sunlight, even though reaching it required more initial growth. This decision-making process seemed to involve something like cost-benefit analysis. The plants were integrating multiple pieces of information, sensory data about their environment, and generating a response that optimized their outcome. They weren't just reacting automatically to the nearest stimulus, they were demonstrating what looks remarkably like choice, even though they possess nothing resembling a brain. The ability to make such decisions relies on plants' sophisticated sensory systems, which are far more extensive than most people realize. Plants can sense light, obviously, They need to detect sunlight for photosynthesis, but their light perception goes far beyond simple light versus dark detection. Plants have multiple types of photoreceptors that detect different wavelengths of light, from ultraviolet through the visible spectrum to far red. They use these receptors not just to find light but to gather detailed information about their environment. For instance, plants can detect the red to far red ratio of light, which changes depending on whether the light has passed through other leaves. When sunlight passes through a leaf canopy, red light gets absorbed for photosynthesis, but far red light passes through relatively unchanged. This means a low red to far red ratio indicates that a plant is being shaded by neighbors. Plants detect this shade signal and respond by elongating their stems, trying to grow taller and overtop their competitors, a strategy called the shade avoidance response. But plants don't just sense light, they also sense gravity, using it to orient their roots downward and shoots upward, a phenomenon called gravitropism. The mechanism involves specialized cells containing dense, starch-filled organelles called statoliths that settle to the bottom of the cell in response to gravity. When the plant is tilted, these statoliths shift position, triggering a cascade of signals that cause growth hormones to redistribute. The result is differential growth. The lower side of a horizontal root grows more slowly than the upper side, causing the root to curve downward, while the opposite occurs in shoots, causing them to curve upward. Plants can also sense touch and mechanical stress, responding to physical contact with changes in growth and development. The response is called thigmomorphogenesis, and it's why plants growing in windy conditions develop shorter, thicker stems than the same species growing in calm air. The mechanical stress of constant movement triggers changes in gene expression that result in a stockier, more stable form. Climbing plants use a similar touch-sensing ability to detect when their tendrils encounter a potential support structure, triggering the tendril to curl around it. Perhaps most surprising, plants can sense sound. In 2012, Monica Gagliano, the same researcher who demonstrated learning in mimosa plants, published research showing that plants respond to specific sound frequencies. She found that corn roots, growing in water, would bend toward the source of a crackling sound at a frequency of 200 to 300 Hz, a frequency similar to the sound that roots themselves make as they grow and crack through soil. This raised the fascinating possibility that plants might be listening to each other's roots, using sound as yet another channel of communication. Further studies found that certain plant species increase their growth rate when exposed to particular sound frequencies, and that plants may produce sound clicks during drought stress, potentially an acoustic distress signal. While this research remains controversial and requires more replication, it suggests that the plant communication network might include dimensions we haven't even begun to fully explore. And so we discovered that plants can learn. Gagliano's 2014 mimosa study showing plants repeatedly dropped onto cushioned surfaces stopped folding leaves after just a few repetitions, having learned the stimulus was harmless. Tested a month later with no drops in between, they still remembered, demonstrating persistent memory without nervous systems, using chemical and electrical signal networks instead of neurons. We learned plants make decisions. Climbing plants presented with multiple supports assessing distance, height, thickness, stability, choosing based on cost-benefit analysis, many selecting distant tall poles over nearby short sticks, optimizing long-term sunlight access rather than simply reaching for nearest support. We discovered plants' sophisticated sensory systems, multiple photoreceptors detecting ultraviolet through far-red wavelengths, sensing red to far-red ratio changes when light passes through leaf canopies. Low ratios indicate shading by neighbors, triggering shade avoidance responsive stem elongation to overtop competitors. We found gravitropism using statoliths, dense starch-filled organelles settling to cell bottoms in response to gravity, shifting when plant tilted, redistributing growth hormones causing differential growth that curves roots downward and shoots upward. Thigmomorphogenesis responding to mechanical stress and touch, wing-grown plants developing shorter, thicker stems, climbing tendrils detecting and curling around supports. We learned Gagliano's 2012 sound research showing corn roots bending toward crackling sounds at 200 to 300 Hz, frequencies similar to sounds roots make cracking through soil, suggesting plants might listen to each other's roots, with some species increasing growth at particular frequencies and producing sound clicks during drought stress. Each sense revealing another layer of plant awareness, another channel of information gathering, another way the green world perceives and responds to its surroundings. Not passive, not simple, but actively sensing, integrating information, adapting with remarkable sophistication. Feel yourself part of this sensing world now, the light and shadow playing across your closed eyelids, gravity gently pulling you down into the earth, the subtle sounds around you creating their own information landscape. The plants doing this constantly, ceaselessly, sensing and responding with elegant complexity. Nothing for you to figure out or understand. Just rest in the awareness that even the stillest, quietest life is rich with perception. As we drift deeper into the remarkable world of plant communication, we arrive at one of the most intricate and strategic relationships in nature, the partnerships plants form with animals, particularly insects, through chemical communication so precise it resembles a coded conversation. We've learned that plants produce volatile organic compounds to warn each other of danger, but this is just the beginning of how they use chemical signals. When a plant is attacked by a specific insect, it can release chemical compounds that attract the natural predators of that insect, essentially calling in reinforcements, recruiting bodyguards from the ecosystem around them. This phenomenon, discovered in the 1990s, is called indirect defense, and it reveals a level of strategic sophistication that challenges our assumptions about plant capabilities. The classic example comes from studies of corn plants attacked by caterpillars. When caterpillar saliva mixes with damaged plant tissue, it triggers a specific chemical response. The corn plant begins releasing a complex blend of volatile compounds that travels through the air, and these compounds attract parasitoid wasps, tiny wasps that lay their eggs inside caterpillars. When the wasp eggs hatch, the larvae consume the caterpillar from the inside, eventually killing it. The plant has essentially sent out a chemical distress call that brings a predator specifically targeting the pest that's attacking it. What makes this even more remarkable is the specificity of the signal. The blend of volatiles released by a caterpillar-damaged plant is different from the blend released by a plant experiencing other types of stress. The wasps can distinguish between these chemical signatures and are attracted specifically to the scent of their host caterpillars feeding. The plant isn't just screaming for help indiscriminately, it's sending a targeted message that says caterpillars are here, which attracts exactly the right predator to solve its problem. Research has shown that more than 30 different plant species use this indirect defense strategy, releasing specific volatile blends that attract predators or parasitoids of their attackers. Lima bean plants release compounds that attract predatory mites when they're infested with spider mites. Wild tobacco plants emit chemicals that attract predatory bugs when attacked by hornworm caterpillars. In each case, the plant's chemical message is tailored to summon the appropriate defender for the specific threat it faces. But the chemical conversation between plants and insects goes beyond just calling for help. Some plants have evolved even more sophisticated strategies involving what researchers call plant-insect manipulation. Certain plants can alter their chemical emissions to manipulate insect behavior in ways that benefit the plant, sometimes recruiting insects as unwitting allies in inter-plant competition. For instance, some plants under attack release volatiles that not only attract predators of their attackers but also repel the pest insects themselves, effectively creating a two-pronged defense: drive away the attackers while simultaneously calling in their enemies. Other plants release compounds that make neighboring plants more attractive to herbivores, essentially redirecting pest insects toward their competitors while making themselves less appealing, a form of chemical deception that benefits the signaling plant at the expense of its neighbors. The relationship between plants and pollinators represents another dimension of plant-insect communication, though one based on cooperation rather than defense. Plants produce elaborate chemical signals, scents, to attract specific pollinators, and the diversity and specificity of these signals is astounding. Some flowers produce scents that mimic the pheromones of female insects, attracting male insects that attempt to mate with the flower and inadvertently transfer pollen in the process. This sexual deception strategy is used by various orchid species and represents an extreme example of chemical communication influencing behavior. The timing of scent production also reveals sophisticated communication strategies. Many flowers release their strongest scents during the hours when their specific pollinators are most active. Night-blooming flowers that rely on moth pollination release intense fragrances primarily after dark, while flowers pollinated by diurnal bees or butterflies concentrate their scent production during daylight hours. This temporal matching of signal production to receiver activity demonstrates that plants don't just broadcast signals randomly, they time their messages to maximize the chance of reaching the intended audience. Beyond insects, plants also communicate chemically with the microbial world and the soil around their roots, a conversation that shapes the plant's health, nutrition, and disease resistance. The root exudates we discussed earlier aren't just detected by other plants, they're also sensed by countless microorganisms in the rhizosphere, the narrow zone of soil directly influenced by root secretions. Plants can selectively recruit beneficial microbes by releasing specific compounds that attract them. When a plant is stressed by disease or nutrient deficiency, it can alter the composition of its root exudates to attract microbes that help address the specific problem. For instance, plants low in nitrogen can release exudates that attract nitrogen-fixing bacteria, which form associations with the roots and convert atmospheric nitrogen into forms the plant can use. Similarly, plants under attack by soil-borne pathogens can recruit antagonistic microbes, bacteria or fungi that suppress or attack the pathogens. This plant-microbe communication is bidirectional. The microbes also send chemical signals to plants, influencing plant growth, development, and immune responses. Certain beneficial bacteria produce compounds that trigger what's called induced systemic resistance in plants, essentially priming the plant's immune system to respond more quickly and effectively to future pathogen attacks. The bacteria are chemically vaccinating the plant, helping it build defenses before disease strikes. And so we discovered indirect defense, plants releasing specific volatile compounds when attacked, recruiting natural predators of their attackers. Corn plants damaged by caterpillar saliva released chemical blends attracting parasitoid wasps that lay eggs inside caterpillars, larvae consuming caterpillars from within. The volatile blend is specific, different from other stress signals, wasps distinguishing chemical signatures and attracted specifically to their host caterpillar scent. We learned over 30 plant species use this strategy, lima beans releasing compounds attracting predatory mites when infested by spider mites, wild tobacco emitting chemicals attracting predatory bugs when attacked by hornworms, each message tailored to summon appropriate defenders for specific threats. We found plants sometimes manipulate insect behavior through chemical deception, releasing volatiles that both repel attackers and attract their predators, a two-pronged defense, or making neighboring plants more attractive to herbivores while becoming less appealing themselves, redirecting pests toward competitors. We discovered orchids producing scents mimicking female insect pheromones, attracting males that attempt mating with flowers and transfer pollen, sexual deception through chemical communication. Scent timing matches pollinator activity, night-blooming flowers releasing fragrances after dark for moths, day-blooming flowers concentrating scent during daylight for bees and butterflies, temporal matching maximizing message delivery. We learned plants recruit beneficial soil microbes through root exudates, nitrogen-deficient plants attracting nitrogen-fixing bacteria, pathogen-attacked plants recruiting antagonistic microbes that suppress attackers. Communication is bidirectional. Beneficial bacteria produce compounds triggering induced systemic resistance, priming plant immune systems to respond faster to future pathogen attacks, chemically vaccinating plants before disease strikes. Each chemical message precisely crafted, timed, targeted to achieve specific outcomes, attracting allies, repelling enemies, manipulating competitors, recruiting microbial partners. The green world engaged in constant chemical conversation with the animal and microbial kingdoms, shaping ecosystems through molecular diplomacy. Feel yourself part of this vast chemical network, breathing in oxygen the plants produce, breathing out carbon dioxide they need, the invisible molecules constantly exchanged, messages and materials flowing between kingdoms of life. Nothing to consciously direct. Just the ancient dance of chemical communication, billions of years in the making, still flowing perfectly all around you. As we continue our peaceful exploration of how plants communicate, we arrive at one of the most profound discoveries in recent plant science, the revelation that plants don't just react to their environment moment by moment, but can anticipate future conditions and prepare for them in advance, demonstrating a form of foresight that seems almost impossible without a brain. This capacity for anticipation was revealed through studies of circadian rhythms in plants, the internal biological clocks that track the 24-hour cycle of day and night. Scientists have known for centuries that many plants move their leaves to track the sun across the sky, a phenomenon called heliotropism, but it wasn't until the 1980s that researchers discovered something remarkable. Plants don't just follow the sun reactively, they anticipate its arrival. In carefully controlled laboratory experiments, researchers observed plants growing under artificial light-dark cycles. Even when the lights were programmed to turn on at the same time each day, plants would begin orienting their leaves toward the direction of the light source 15 to 30 minutes before the lights actually came on. the plants were using their internal clock to predict when dawn would arrive and positioning themselves in advance to maximize photosynthesis the moment light became available. This anticipatory behavior demonstrates that plants possess something functionally equivalent to memory of past patterns and the ability to use that memory to predict future events. They're not simply responding to current stimuli, they're integrating information over time and making preparations based on learned patterns. When researchers disrupted the light schedule, presenting dawn at unexpected times, the plants initially were caught unprepared, but within a few days, they had adjusted their anticipatory movements to the new schedule, essentially learning the new pattern and forecasting accordingly. The circadian clock that enables this prediction isn't just about tracking light and dark, it coordinates thousands of biochemical processes throughout the plant, ensuring that the right genes are activated at the right times of day. Plants turn on genes for photosynthesis just before dawn, activating the molecular machinery needed to capture sunlight before the light actually arrives. They activate defensive chemical production during the hours when insect herbivores are most likely to feed. They time their flower opening to coincide with the active hours of their pollinators. Everything is scheduled, coordinated to the rhythms of day and night. Even more remarkably, plants can integrate information about seasonal changes and use it to time major life events like flowering, leaf drop, or dormancy. They measure day length, specifically the ratio of light hours to dark hours, through specialized photoreceptor proteins that can essentially count how long the light lasts each day. This process, called photoperiodism, allows plants to track the seasons with remarkable precision. Short-day plants, which flower when nights become longer than a critical threshold, are actually measuring night length rather than day length. If you interrupt the dark period with even a brief flash of light, you can prevent flowering entirely, because the plant interprets the light flash as breaking the night into two shorter periods, neither long enough to trigger flowering. This demonstrates that plants are not just passively recording day length but actively processing this information according to specific rules, essentially performing calculations to determine whether conditions are right for flowering. This ability to track seasons and anticipate future conditions extends to stress preparation. Research has shown that plants can acclimate to predictable stresses before they occur. For instance, plants growing in environments with regular cold nights develop frost resistance before the temperature actually drops, using the shortening day length of autumn as a cue that winter is approaching. They don't wait to experience frost and then react. They prepare in advance based on seasonal signals, increasing production of antifreeze proteins and protective compounds weeks before the first freeze. Plants also demonstrate remarkable transgenerational communication, the ability to pass information from one generation to the next in ways that go beyond standard genetic inheritance. This process, called epigenetic inheritance, allows plants that have experienced stress to chemically modify their DNA in ways that make their offspring better prepared to handle similar stress, even if the offspring never experienced that stress directly during their own development. For example, parent plants exposed to drought conditions can produce offspring with enhanced drought tolerance, thicker cuticles, deeper root systems, more efficient water use, even when those offspring are grown in well-watered conditions. The parent plant has essentially sent a message to the next generation saying drought is likely in this environment, here are adaptations you'll need. And this message is encoded in chemical modifications to DNA called methylation that affect how genes are expressed without changing the actual DNA sequence. Similarly, plants attacked by herbivores can produce offspring with enhanced defensive capabilities. Studies with wild radish plants showed that parents that had been heavily browsed by caterpillars produced offspring that were less palatable to caterpillars and more attractive to the parasitoid wasps that attack caterpillars. The offspring inherited both enhanced direct defenses and enhanced indirect defenses, even though they had never encountered a caterpillar themselves. The parent plants were preparing the next generation for threats based on their own experience. And so we discovered plants can anticipate the future. 1980s laboratory experiments revealing plants oriented leaves toward light source 15 to 30 minutes before lights turned on, using internal clocks to predict dawn and maximize photosynthesis the moment light arrived. This demonstrates memory of past patterns and ability to predict future events, not merely reacting to current stimuli but integrating information over time. We learned circadian clocks coordinate thousands of biochemical processes, turning on photosynthesis genes just before dawn, activating defensive chemicals when herbivores most likely feed, timing flower opening to pollinator activity hours, everything scheduled to day-night rhythms. We discovered photoperiodism, plants measuring day length through photoreceptor proteins that count light hours. Short day plants actually measure night length, brief light flash during dark period preventing flowering by breaking night into two short periods, demonstrating active information processing and calculation rather than passive recording. We found plants acclimate to predictable stresses before they occur, shortening autumn days triggering frost resistance weeks before first freeze, producing antifreeze proteins and protective compounds in advance based on seasonal signals rather than waiting to experience frost then reacting. We learned of epigenetic inheritance, transgenerational communication where stress-experienced parents chemically modify DNA through methylation, producing offspring better prepared for similar stress even without experiencing it directly. Drought-exposed parents produce offspring with enhanced drought tolerance, thicker cuticles, deeper roots, efficient water use, even when offspring grown in well-watered conditions. We discovered herbivore-attacked wild radish plants produce offspring less palatable to caterpillars and more attractive to parasitoid wasps, inheriting enhanced direct and indirect defenses without encountering caterpillars themselves, parents preparing next generation for threats based on experience. Each discovery revealing temporal intelligence, plants tracking past patterns, predicting future conditions, preparing in advance, even sending messages across generations. Not trapped in the present moment but existing across time, remembering yesterday, anticipating tomorrow, investing in generations yet to come. Feel yourself existing in time like these plants, rooted in this present moment but connected to all the moments that came before and all that will come after. The rhythm of day and night, season following season, generation after generation, the ancient patterns still pulsing through all living things. Nothing to grasp or hold. Just the natural flow of time, carrying you gently forward, preparing you for tomorrow's rest even as today's rest begins. As we drift deeper into the remarkable world of plant communication, we arrive at discoveries that reveal just how actively plants engineer their environments, shaping the communities around them through chemical signals that influence not just their immediate neighbors, but entire ecosystems. One of the most fascinating examples of this ecosystem engineering involves a phenomenon called plant-soil feedback, where plants actively modify the microbial community in the soil around their roots, and those microbes, in turn, affect which plants can successfully grow in that soil in the future. Each plant species cultivates a unique microbial community through its specific blend of root exudates, essentially creating a customized soil environment that either favors its own species or favors different species, depending on the ecological strategy. Research has shown that when the same plant species grows in the same soil repeatedly, it often accumulates species-specific pathogens, harmful microbes that specialize in attacking that particular plant. This creates negative plant-soil feedback, where the soil becomes increasingly hostile to that species over time. This is one reason why crop rotation is so effective in agriculture. Planting different crops in succession prevents the buildup of species-specific pathogens, breaking the negative feedback cycle. But it's also why, in natural forests, you rarely find young trees growing directly beneath their parent tree. The soil under a mature tree has accumulated pathogens specific to that species, making it a poor place for its own offspring to establish, even though it would seem like the ideal location with access to the parent's mycorrhizal network. However, some plants create positive plant-soil feedback, cultivating microbial communities that benefit their own species. Invasive plants are often particularly good at this, which helps explain their success in new environments. Garlic mustard, an invasive plant in North American forests, produces root exudates that suppress mycorrhizal fungi that most native plants depend on, while garlic mustard itself doesn't need mycorrhizal partners. By chemically disrupting the fungal networks that native plants rely on, garlic mustard creates soil conditions that favor itself at the expense of the native plant community. This active modification of soil communities extends to influencing nutrient cycling throughout the ecosystem. Some plants produce root exudates that stimulate microbial decomposition of organic matter, essentially accelerating the release of nutrients from dead material into forms that plants can absorb. Other plants produce exudates that slow decomposition, effectively locking nutrients away in organic matter. By controlling the rate of decomposition through their chemical signals to soil microbes, plants can influence nutrient availability not just for themselves but for the entire plant community. The chemical communication between plants also extends above ground in ways that structure entire communities. In dense forests, the chemical signals exchanged between trees can coordinate behavior at the community level. Research in tropical forests has found that some tree species, when attacked by herbivores, release volatile compounds that trigger defensive responses not just in the same species but across multiple different species in the vicinity. This creates a neighborhood-wide defensive alert system where different species essentially cooperate in defense, even though they're competing for light, water, and nutrients. This cooperation becomes even more remarkable in the context of what researchers call defense synchrony. Studies have shown that when one individual of a particular species is attacked, other individuals of the same species throughout the forest, sometimes hundreds of meters away, increase their defensive chemical production within days, even when they're too distant to detect airborne signals from the attacked individual. The signal appears to travel through the mycorrhizal network, creating a species-wide defensive coordination that operates at the scale of the entire forest. But plants don't just defend themselves and their communities, they can also actively suppress competitors through allelopathic chemicals that we touched on earlier. Some plants have evolved remarkably sophisticated allelopathic strategies. The black walnut we mentioned produces juglone, but research has revealed that the production and release of this compound is actually regulated based on the presence of competitors. Black walnut trees produce more juglone when growing near other species, suggesting they can somehow sense competitive pressure and respond by increasing their chemical warfare. Even more intriguing, some plants appear to target their allelopathic compounds to specific competitors. Spotted knapweed, another invasive plant, produces a chemical called catechin that is particularly toxic to North American native plants but less harmful to plants from its native Eurasian range. This selective toxicity may give it an advantage in its invaded range, where native plants have an evolved resistance to catechin, while in its home range, co-evolved plants are less affected. The plant is essentially using chemical weapons that work better against the species it encounters in its new environment than against the species it evolved alongside. Research into plant communication has also revealed that plants can distinguish between self and non-self. They can recognize their own tissues versus tissues of other plants, even other plants of the same species. This self-recognition ability has been demonstrated most clearly in parasitic plants like dodder, which must find and attach to host plants to survive since they cannot photosynthesize on their own. Dodder seedlings, when they encounter potential hosts, can actually smell them, detecting the volatile organic compounds different plant species emit, and they show preferences, growing preferentially towards species that will make better hosts. Even more remarkably, when dodder encounters its own species versus a different species, it can distinguish between them and will preferentially parasitize the other species rather than its genetic relatives. This kin recognition in a parasitic plant suggests a sophisticated ability to process chemical information about identity and make strategic decisions based on genetic relationships. And so we discovered plant-soil feedback, each species cultivating unique microbial communities through specific root-exudate blends, creating customized soil environments. Negative feedback accumulates species-specific pathogens over time, making soil hostile to that species, explaining why young trees rarely establish under parents and why crop rotation prevents pathogen buildup. We learned invasive plants like garlic mustard create positive plant-soil feedback, producing exudates suppressing mycorrhizal fungi native plants need while garlic mustard doesn't require fungal partners, chemically disrupting networks natives depend on, creating conditions favoring itself. We found plants control nutrient cycling through root exudates, some stimulating microbial decomposition to accelerate nutrient release, others slowing decomposition to lock nutrients in organic matter, influencing availability for entire communities. We discovered tropical forest trees attacked by herbivores release volatiles triggering defensive responses across multiple different species, neighborhood-wide alert system where competing species cooperate in defense. Defense synchrony shows individuals of same species hundreds of meters apart increase defensive production within days via mycorrhizal network signals, creating species-wide coordination at forest scale. We learned black walnut trees produce more juglone when growing near competitors, sensing competitive pressure and responding with increased chemical warfare. Spotted knapweed produces catechin particularly toxic to North American natives but less harmful to Eurasian plants from its native range, selective toxicity giving advantage in invaded range where natives lack evolved resistance. We discovered plants distinguish self from non-self. Dodder parasitic plants smell potential hosts' volatile compounds, showing preferences for better hosts. Dodder can distinguish own species from others, preferentially parasitizing non-relatives rather than genetic kin. Kin recognition demonstrating sophisticated chemical information processing and strategic decision-making based on genetic relationships. Each discovery revealing plants as active ecosystem engineers, not passive recipients of conditions but shapers of communities, modifiers of soil, coordinators of defense, wielders of selective chemical weapons, recognizers of kin. The green world engaged in constant chemical diplomacy and warfare, cooperation and competition, all without brains, without nerves, without consciousness as we know it, yet with results that look remarkably intelligent. Feel yourself part of this chemical web, your breath connected to their photosynthesis, your body built from elements they helped cycle through soil and air. The boundaries between organisms blurring when you zoom out far enough, all life interconnected through chemical conversations older than consciousness itself. Nothing to figure out, nothing to solve. Just rest in this ancient network of connection. As we continue our peaceful journey through the world of plant communication, we arrive at some of the most recent and revolutionary discoveries, findings that are reshaping how we understand plant intelligence, memory, and even the possibility of something resembling consciousness in organisms without brains. One of the most profound areas of recent research involves plant electrical signaling, the discovery that plants generate and transmit electrical signals through their tissues in ways that are functionally similar to nerve impulses in animals, even though plants have no nervous system. These electrical signals were first documented in the 1800s, but it wasn't until the late 20th and early 21st centuries that scientists began to understand their complexity and importance. Plants generate electrical potentials across their cell membranes, just as animal neurons do, and when a plant is stimulated by touch, damage, light, temperature change, or chemical signals, these electrical potentials can change rapidly and propagate through the plant's tissues. These traveling electrical signals, called action potentials in animals, work through similar mechanisms in plants. Ion channels in cell membranes open and close, allowing charged particles like calcium, potassium, and chloride to flow in and out, creating waves of electrical change that move from cell to cell. The speed at which these signals travel varies depending on the plant and the type of signal, but some can move remarkably fast, up to 1 meter per second in certain plants. The Venus flytrap provides one of the most dramatic examples. When an insect touches the trigger hairs inside the trap, it generates an electrical signal that propagates across the leaf surface. The trap counts these signals. It requires 2 signals within about 20 seconds to close, essentially performing a calculation to avoid wasting energy closing on raindrops or debris. Once the threshold is reached, the electrical signal triggers a rapid change in cell water pressure that snaps the trap shut in less than 1/10 of a second, one of the fastest movements in the plant kingdom. But electrical signaling isn't limited to carnivorous plants with obvious movements. All plants appear to use electrical signals to coordinate responses across their bodies. When a leaf is damaged, electrical signals can travel through the stem to other leaves, triggering defensive responses throughout the plant. When roots encounter drought, electrical signals can travel upward to leaves, causing stomata, the tiny pores that control gas exchange, to close, conserving water. The plant is using electrical signaling to communicate internally, coordinating the activities of different parts of its body just as your nervous system coordinates your organs, but without centralized control or a brain. This electrical signaling system also allows plants to integrate information from multiple senses and generate complex, context-dependent responses. Research has shown that plants don't respond to stimuli in isolation but rather integrate multiple signals, light, temperature, moisture, chemical signals from neighbors, mechanical stress, and generate responses that depend on the combination of all these inputs. A plant might respond differently to the same chemical signal depending on the time of day, the amount of light available, whether it's already under drought stress, and what signals it's receiving from neighboring plants through the soil. This integration of multiple information streams and context-dependent decision-making is sometimes called plant cognition, a term that remains controversial because it seems to imply consciousness or awareness. But researchers who study plant behavior argue that if we define cognition functionally, as the ability to acquire information, process it, store it, and use it to generate adaptive behavior, then plants clearly display cognitive abilities, even if the mechanisms are entirely different from animal cognition and don't involve anything like conscious experience. One of the most intriguing aspects of plant cognition is the phenomenon of habituation we discussed earlier with Monica Gagliano's mimosa experiments, but recent research has revealed that plant learning and memory extend far beyond simple habituation. Plants appear capable of associative learning, the same type of learning Ivan Pavlov famously demonstrated with dogs, where an initially neutral stimulus becomes associated with a meaningful stimulus through repeated pairing. In 2016, Monica Gagliano published research showing that pea plants could learn to associate a light source with airflow. She grew pea plants in Y-shaped mazes where they had to choose which direction to grow. Initially, a fan created airflow from one direction while light came from a different direction. Pea plants naturally grow toward light, but Gagliano arranged the experiment so that whichever direction the airflow came from, that's where the light would be during the actual test, even though during training the light and air came from different directions. After several training sessions where light and airflow were paired but coming from different locations, Gagliano tested the plants with airflow alone, no light. The plants grew toward the direction of the airflow, even though airflow by itself is not normally something plants grow toward. They had learned to associate the airflow with the presence of light in that direction, essentially forming a Pavlovian association between two environmental cues. This demonstrated that plants can not only modify their behavior based on experience but can learn relationships between different environmental signals and use those learned relationships to predict future conditions. The implications of this research are profound. If plants can learn associations between unrelated stimuli, it suggests they possess more sophisticated information processing abilities than previously recognized. They're not just responding to current stimuli but are building models of their environment, learning which signals predict other signals, which conditions tend to occur together, which responses work in which contexts. This capacity for learning and memory raises questions about whether plants might experience something analogous to stress or suffering. While plants almost certainly don't experience anything like human or animal consciousness, they lack the centralized nervous systems that seem necessary for subjective experience. They do have sophisticated systems for detecting and responding to damage, stress, and threat. They generate electrical signals in response to injury. They produce stress hormones like jasmonic acid and ethylene. They remember past attacks and prepare for future ones. Some researchers argue that we should recognize plant suffering not as conscious pain but as a form of biological harm that matters morally, similar to how we might consider that damaging an ecosystem harms it even though ecosystems don't have consciousness. Others argue that without consciousness, there can be no morally relevant suffering, and that plants' sophisticated responses to damage are purely mechanistic, no more indicative of suffering than a computer's response to a virus is indicative of computer suffering. And so we discovered plant electrical signaling, action potentials generated across cell membranes through ion channel opening and closing, creating waves of electrical change moving from cell to cell, some traveling up to 1 meter per second. Venus flytraps count electrical signals from trigger hairs, requiring 2 signals within 20 seconds before closing, performing calculation to avoid wasting energy, traps snapping shut in less than 1/10 second, among fastest plant movements. We learned all plants use electrical signals to coordinate responses, damaged leaves sending signals through stems to other leaves triggering defensive responses, drought-encountering roots sending signals upward causing stomata to close conserving water, internal communication coordinating body parts without centralized control. We discovered plants integrate multiple information streams, light, temperature, moisture, chemical signals, mechanical stress, generating context-dependent responses. Same chemical signal produces different responses depending on time of day, light availability, drought stress status, neighboring plant signals, demonstrating complex information integration. We explored plant cognition debate, functionally defined as acquiring, processing, storing, and using information to generate adaptive behavior. Plants clearly display cognitive abilities through different mechanisms than animals, though without conscious experience. We learned Gagliano's 2016 Plant Associative Learning Study using Y-shaped mazes, plants trained with paired airflow and light, from different directions, then tested with airflow alone grew toward airflow direction, having learned airflow predicts light location. Pavlovian association between environmental cues demonstrating plants learn relationships between unrelated stimuli and use learned relationships to predict future conditions. We found associative learning suggests plants build environmental models, learning which signals predict others, which conditions occur together, which responses work in which contexts, more sophisticated information processing than previously recognized. We considered whether plants might experience stress or suffering, debate between recognizing biological harm that matters morally even without consciousness versus arguing only conscious suffering has moral relevance, plants' sophisticated damage responses being purely mechanistic like computer virus responses. Each discovery pushing the boundaries of what we mean by intelligence, learning, cognition, raising profound questions about consciousness, suffering, moral consideration. The green world revealing depths of complexity that challenge our categories, our definitions, our understanding of what it means to be aware, to know, to respond to the world. Feel yourself resting among beings whose inner lives we're only beginning to glimpse, whose ways of knowing are so different from ours we barely have language for them. Nothing to resolve tonight, nothing to conclude. Just rest in the mystery, in the profound strangeness of minds so different from ours yet solving similar problems, sensing, learning, remembering, deciding, through such different pathways. As we approach the completion of our journey through the hidden world of plant communication, we arrive at some of the most recent and forward-looking discoveries, findings that are revealing entirely new dimensions of how plants interact with the world and pointing toward future breakthroughs we're only beginning to imagine. One of the most exciting areas of current research involves understanding how plants use sound not just to respond to vibrations, as we discussed with Monica Gagliano's work on root acoustics, but potentially to communicate actively with each other through substrate-borne vibrations. In 2019, researchers discovered that when certain plants are stressed by drought, they emit ultrasonic clicks, sounds at frequencies too high for human hearing but detectable with sensitive microphones. These ultrasonic emissions appear to be produced by cavitation, the formation and collapse of air bubbles in the plant's water-conducting vessels as the water column breaks under tension during severe drought stress. The frequency and intensity of these clicks increase as drought stress intensifies, essentially creating an acoustic signature of the plant's stress level. What makes this particularly intriguing is that neighboring plants might be able to detect these sounds and use them as early warning signals, allowing them to activate their own drought response mechanisms before they experience water stress themselves. Research teams are now investigating whether plants can distinguish between different types of stress-induced sounds, drought clicks versus sounds produced by insect chewing versus wind damage, and whether they respond differently to these distinct acoustic signatures. If plants can indeed use sound as an information channel, detecting and interpreting acoustic signals from their environment, it would add yet another layer to the already complex network of plant communication we've explored tonight. Beyond sound, scientists are discovering that plants can detect and respond to incredibly subtle environmental cues that we're only beginning to measure. Recent research has shown that some plants can sense the Earth's magnetic field and use it for orientation, though the mechanisms and functions are still being investigated. Even more remarkably, there's emerging evidence that plants might be able to sense the presence of water at a distance, growing their roots preferentially toward moisture sources even before the roots encounter any gradient in soil moisture that could be detected through normal sensory mechanisms. In 2017, Monica Gagliano published research suggesting that pea plant roots could detect the sound of running water and would grow toward the source of that sound, even when the water was sealed inside a pipe and there was no moisture gradient in the soil. The plants appeared to be responding to the acoustic vibrations of flowing water, essentially hearing water and growing toward it. While this research remains controversial and requires further replication, it suggests that plants' sensory world might be far richer than we currently understand. The sophistication of plant sensory systems has led some researchers to investigate whether plants might have something analogous to the different senses being integrated in a central location, not a brain, but perhaps a coordination center that combines information from different sources to generate unified responses. Some scientists have proposed that the root tip functions as a kind of command center for the plant, integrating signals from the environment and coordinating growth responses. The root tip contains a region called the transition zone, where cells are transitioning from active division to elongation, and this zone shows remarkable electrical activity and sensitivity to multiple signals, gravity, light. Even though the root is underground, light can penetrate through soil particles, touch, chemicals, moisture, nutrients, temperature, and potentially sound and magnetic fields. Some researchers have argued that this root tip transition zone, with its integration of multiple signal types and its role in directing growth, serves functions analogous to those of a simple brain, though without consciousness or centralized processing as animals have. This concept, sometimes called the root-brain hypothesis, remains highly controversial. Critics argue that the analogy to brains is misleading because plants don't have neurons, action potentials work differently in plants than in animals, and there's no evidence of centralized information processing. Proponents counter that if we focus on function rather than mechanism, on what the system does rather than how it does it, then the root tip's integration of multiple sensory inputs to coordinate adaptive growth responses is performing computations similar to what brains do, even if through entirely different means. Regardless of whether we call it a brain, the root tip's ability to sense and integrate information is remarkable. Roots can detect obstacles in their path and grow around them, can sense the presence of roots from neighboring plants and adjust their growth pattern, growing aggressively toward resources when near competitors or growing more conservatively when near kin. They can sense gradients of nutrients in the soil and adjust their growth to maximize contact with nutrient-rich patches while minimizing investment in nutrient-poor areas. All of this requires constant sensing, information processing, and growth adjustment. This leads to one of the most profound questions in plant science. How do plants make decisions without a brain? We've seen throughout this journey that plants make what appear to be choices: which direction to grow, when to flower, how much to invest in defense versus growth, whether to cooperate or compete with neighbors. But if there's no central decision maker, no executive function, no consciousness weighing options, how do these decisions emerge? The current understanding is that plant decision-making is distributed. It emerges from the collective behavior of millions of cells, each responding to local signals and conditions according to simple rules, but with the aggregate result being sophisticated, adaptive behavior at the level of the whole plant. It's similar to how ant colonies make complex decisions about where to build nests or which food sources to exploit, even though no individual ant understands the overall situation. The intelligence is collective, emergent, arising from many simple interactions rather than from centralized planning. This distributed intelligence means that damaging part of a plant doesn't necessarily prevent it from making decisions, unlike damaging a brain in an animal. Plants can be cut in half, lose branches or roots, and the remaining portions continue to sense their environment and coordinate adaptive responses. Each part retains decision-making capacity because the intelligence isn't localized but distributed throughout the organism. Understanding plant communication and intelligence has practical implications that extend far beyond academic curiosity. Researchers are now exploring how to use plants' own communication systems to improve agriculture, making crops more resistant to pests and diseases by enhancing their natural chemical communication networks. Some scientists are investigating whether we could artificially trigger plant defensive responses by exposing crops to the volatile compounds that normally serve as warning signals, essentially vaccinating the plants against future attack without using pesticides. And so we discovered plants emit ultrasonic clicks during drought stress, cavitation sounds from air bubbles forming and collapsing in water-conducting vessels, frequency and intensity increasing with stress severity. Neighboring plants might detect these acoustic signatures as early warnings, activating drought responses before experiencing water stress themselves. Researchers investigating whether plants distinguish drought clicks from insect chewing sounds or wind damage. We learned recent research shows some plants sense Earth's magnetic field for orientation. Emerging evidence suggests plants might detect water at distance, growing roots toward moisture sources before encountering soil moisture gradients. Gagliano's 2017 P-root study suggesting roots grow towards sound of running water in sealed pipes, appearing to hear water through acoustic vibrations. We explored the root-brain hypothesis, root tip transition zone showing remarkable electrical activity and sensitivity to gravity, light penetrating soil, touch, chemicals, moisture, nutrients, temperature, potentially sound and magnetic fields. Some researchers argue this integration of multiple signal types directing growth serves functions analogous to simple brains, though critics counter plants lack neurons and centralized processing, analogy misleading. We discovered root tips sense obstacles and grow around them, detect neighboring plant roots adjusting growth patterns, aggressive toward competitor resources, conservative near kin, sense nutrient gradients maximizing contact with nutrient-rich patches, requires constant sensing, information processing, growth adjustment. We learned plant decision-making is distributed, not centralized, emerges from millions of cells each responding to local signals with simple rules, aggregate result being sophisticated adaptive behavior, similar to ant colony intelligence arising from many simple interactions without centralized planning. Distributed intelligence means damaged plants continue making decisions, each part retaining capacity since intelligence isn't localized. We found practical implications, researchers exploring use of plants' communication systems to improve agriculture, triggering defensive responses by exposing crops to volatile warning compounds, essentially vaccinating plants against future attack without pesticides. Each discovery revealing new dimensions of plant sensing, communicating, deciding, the boundaries of what we thought possible constantly expanding as methods improve and questions deepen. The green world more complex, more capable, more interconnected than we imagined even decades ago. Feel yourself part of this distributed intelligence, this networked knowing, cells in your body each responding to local signals, the aggregate creating you, your consciousness emerging from countless simple interactions. Plants and people, different pathways to similar solutions, sensing, responding, adapting, surviving. Rest now in this kinship, this shared biology of information and response. As we drift toward the completion of our journey through the hidden world of plant communication, let's gather the final threads, the discoveries that remind us how profoundly we've underestimated the green world, and how much mystery still remains in the quiet lives of plants. Throughout this journey, we've witnessed a revolution in understanding, from seeing plants as passive organisms simply existing in their environment, to recognizing them as active communicators, sophisticated sensors, and in some ways, intelligent beings that perceive, remember, learn, and make decisions, all without anything resembling a brain. We've learned that communication is the foundation of plant life, that no plant exists in isolation, but rather as a node in vast networks, chemical networks carried through air, fungal networks threading through soil, electrical networks pulsing through tissues, acoustic networks vibrating through water and earth, all overlapping, all carrying information, all enabling the coordination and cooperation that allows plants to thrive. But perhaps the deepest lesson from plant communication science is how it challenges our assumptions about what intelligence requires, what awareness requires, what it means to sense and respond to the world. For most of human history, we assumed that meaningful perception, that learning and memory, that decision-making and problem-solving, all required centralized nervous systems, required brains, perhaps required something like consciousness. Plants have demonstrated that these assumptions were wrong. They perceive their environment with exquisite sensitivity, detecting not just light and gravity and touch, but chemical signals from neighbors, electrical impulses from fungal partners, potentially even magnetic fields and sound vibrations we're only beginning to measure. They learn from experience, forming memories that persist for weeks or months, modifying their behavior based on past events and using those memories to predict future conditions. They make decisions, complex ones, integrating multiple streams of information, assessing costs and benefits, choosing between options in ways that optimize their survival and reproduction. They communicate, sending warning signals to kin, recruiting allies against enemies, manipulating competitors, negotiating with partners, all through chemical languages of remarkable sophistication and specificity. And they do all of this without a single neuron, without a brain, without anything we recognize as consciousness. Their intelligence is distributed, emergent, arising from the collective behavior of millions of cells, each responding to local conditions, creating coordinated responses at the scale of the whole organism, the whole forest, the whole ecosystem. This distributed intelligence, this consciousness without centralization, challenges us to expand our definitions, to recognize that there are many ways to be intelligent, many ways to process information and generate adaptive behavior, many ways to exist as a knowing being in the world. It also invites us to reconsider our relationship with the plant world. These are not objects, not mere resources, not the backdrop against which animal life plays out. They are participants, active agents in ecosystems, communicators in vast networks of information exchange, organisms with their own forms of memory and learning, beings whose inner lives we're only beginning to glimpse. Some researchers now speak of plant neurobiology, plant cognition, even plant intelligence, terms that make some scientists uncomfortable because they seem to anthropomorphize, to project human qualities onto organisms fundamentally different from us. But other researchers argue that these terms, if properly defined, capture something real about what plants do, that the functional similarities between plant behavior and animal cognition, despite the mechanistic differences, deserve recognition. The debate continues, and perhaps that's appropriate. Perhaps we shouldn't rush to conclusions about plant minds, plant awareness, plant experience. What matters is that we've learned to see them differently, to recognize the complexity hidden beneath the apparent simplicity, to appreciate the constant communication flowing through forest and field, garden and meadow, communication we cannot hear with our ears, but which science is teaching us to detect and decode. One of the most beautiful aspects of plant communication research is how it reveals interconnection as fundamental to life. Plants cannot exist alone. They depend on fungi to extend their reach into soil, on bacteria to fix nitrogen and defend against pathogens, on insects for pollination and seed dispersal, on neighboring plants for warning signals and resource sharing. The rugged individual, the solitary survivor, is a fiction. Life is networked, communal, cooperative at its core. And we are part of these networks too. The oxygen we breathe comes from plant photosynthesis. The food we eat grows from soil communities that plants cultivate. The climate that makes Earth habitable is maintained partly by plant transpiration and carbon sequestration. When we walk through a forest, we're walking through an active communication network, chemical signals flowing around us, electrical pulses running beneath our feet, fungal threads connecting everything below the surface, and we are affecting that network simply by being present, our footsteps sensed by roots, our breath contributing carbon dioxide that plants will use, our presence noted in ways we're only beginning to understand. This recognition of interconnection, a fundamental relationship between all living things, is perhaps the deepest gift of plant communication science. It reminds us that separation is illusion, that boundaries between organisms are porous, that individuals are always embedded in communities, and communities in ecosystems, and ecosystems in the biosphere, all communicating, all exchanging materials and energy and information, creating the living planet we call home. And so, dear listener, we've completed our journey through 100 facts about plant communication, from the first discoveries in 1983. That damaged trees send airborne warnings to their neighbors, through the revelation of the Wood Wide Web, the underground fungal network connecting forest communities, to the latest findings about plant learning, plant memory, plant acoustics, and the ongoing debates about plant intelligence and cognition. We've learned of volatile organic compounds carrying messages through air, of root exudates creating chemical signatures in soil, of electrical signals coordinating responses within plants, of kin recognition allowing plants to distinguish relatives from strangers, of transgenerational communication preparing offspring for threats parents experienced, of sound production during stress and potential acoustic sensing. We've discovered plants anticipating dawn before it arrives, tracking seasons through photoperiodism, making cost-benefit calculations about where to grow, recruiting predatory insects to defend against herbivores, cultivating beneficial microbes while suppressing pathogens, coordinating forest-wide defensive responses through mycorrhizal networks, manipulating competitors through allelopathic warfare, and doing all of this through distributed intelligence that emerges from millions of cells following simple local rules. Each fact revealing another layer of complexity, another dimension of capability, another reason to regard the green world with wonder and respect. Plants are not less alive than animals, just differently alive, solving the challenges of existence through other means, perceiving through other senses, communicating through other languages, but achieving outcomes that look remarkably sophisticated when we finally learn to see them. But the facts are just the beginning. What matters is the shift in perspective, the recognition that consciousness and intelligence come in many forms, that communication happens through countless channels, that the apparently silent green world is actually engaged in constant conversation, chemical and electrical and perhaps acoustic and magnetic, a conversation we're only beginning to understand. As you lie here tonight, drifting towards sleep, know that you're surrounded by this conversation. The houseplants in your room, if you have them, are sensing the darkness, adjusting their circadian clocks, perhaps even sensing your presence through the carbon dioxide you exhale. The trees outside are connected underground through fungal networks, sharing resources, exchanging warnings, coordinating their seasonal rhythms. Even in a city, even indoors, the plant world's influence touches you, through the oxygen in the air, the wood in your furniture, the cotton in your sheets, the food in your stomach, all gifts from the photosynthetic world, from organisms that harvest light and turn it into life. There's nothing you need to do with this knowledge, nothing you need to remember or act upon. Just let it settle into your awareness like leaves settling to forest floor, becoming part of the rich substrate from which new growth emerges. Feel yourself part of this living planet, breathing in what plants breathe out, breathing out what plants breathe in, the fundamental exchange that makes Earth habitable, the chemical conversation between plant and animal that has sustained life for hundreds of millions of years, and continues now, with every breath you take. Your body is made of plant matter, carbon that was once captured from air through photosynthesis, water that passed through plant tissues, minerals that were cycled through soil communities that plants cultivate. You are not separate from the plant world. You are built from it, sustained by it, embedded in networks of exchange that flow through all living things. And as you sleep, as your consciousness fades and your breathing deepens, the plants continue their work, photosynthesizing if there's light, respiring always, communicating through their chemical networks, sensing and responding and adjusting, the green conversation continuing through the night, patient, persistent, fundamental to the living earth. Rest now, in the knowledge that you're cradled by this living planet, supported by systems of communication and cooperation more ancient than consciousness, more fundamental than thought, embedded in networks that connect all life, plant and animal, fungus and bacteria, all communicating, all cooperating, all part of the vast experiment of life on Earth. Thank you for journeying through the hidden world of plants with me tonight, for learning these 100 facts about how the silent ones speak, for opening your mind to forms of intelligence so different from your own, for staying until the end, or for drifting away whenever sleep called you. Either way, exactly right. May your sleep be deep. May your dreams be peaceful. May you wake tomorrow with new eyes for the green world, seeing the communication flowing through every garden and forest, every field and meadow, the constant chemical conversation that sustains life on Earth. May you rest knowing that intelligence takes many forms, that awareness doesn't require consciousness, that the capacity to sense and respond and remember and learn exists in kingdoms of life very different from your own, and that this diversity of ways of being, this multitude of solutions to the challenge of existence, makes our planet more resilient, more beautiful, more wondrous. Sleep well, dear listener, in the embrace of the green world, knowing you are not observer but participant, not separate but embedded, not above but among the countless forms of life that sense and signal, communicate and cooperate, making Earth the living marvel it is. Good night, rest deeply, wake gently, to a world more alive, more aware, more interconnected than you imagined, a world where even the quietest beings are speaking, if we only learn their language.